Hello, my name is Katherine O'Meara and I am a part of the class of 2023. My abstract's title is The Clinical Significance of Elevated NT-proB-type natriuretic peptide levels in children with creatine transporter deficiency. My abstract is accepted to NCUR 2020. I conducted this study as a 2019 summer intern at the National Institutes of Health through the National Institute of Child Health and Human Development. The study focused on BNP, or B-type natriuretic peptide levels, within the population of children with creatine transporter deficiency who are a part of the National Institutes of Health natural history study of the disease. Creatine transporter deficiency is a genetic disorder that primarily affects the brain. It is caused by a mutation in the SLC6A8 gene, which provides instructions for making a protein that transports creatine into cells. Creatine transporter deficiency is an X-linked mutation, which means that the disease predominantly affects males. Creatine is needed for the body to store and use energy properly. This disorder usually causes intellectual disability and behavioral disorders, as well as slow growth and delayed development of motor skills. In this population, cardiomyopathy is more prevalent than normal populations as the heart needs creatine to function properly. BNP is a clinical value found in blood that is used as a diagnostic tool for adult cardiomyopathy. BNP is a counter-regulatory hormone, meaning it works against natural forces in the body. BNP is produced primarily by the ventricles of the heart and released in response to increased stretching of the heart or increased stress on the heart. It works by removing sodium and water to help reduce blood pressure as well as dilating the blood vessels. Compared to the adult reference ranges for BNP, about 60%, or 12 out of 21, of the National Institute of Health's subjects had elevated BNP levels. Currently, there's no standard reference range for pediatrics, but there was a study review conducted of 4 different studies that made a proposed reference range for pediatric BNP levels. Using this range, only about 19%, or 4 out of 21, of the children had high BNP levels. Of those 4, the subject with the highest level also had a more recent level within the proposed normal range. Thus, the clinical significance couldn't be completely determined, but my work will not only influence future decision-making within the research group, as well as help alleviate parents' worries if their child's creatine level is above the adult reference range. Further research would need to be conducted to determine a standard range for pediatric BNP levels. I hope you enjoyed my abstract. Thank you.